Hello. Good afternoon, everybody. This is Martin McGlynn. I'm the President and CEO of StemCells, Inc. I would like to thank you all for joining our webcast today. Before going any further, I would just like to mention that during today's call, we will be making some forward-looking statements which reflect our current views and are based upon certain assumptions that may or may not ultimately prove valid. We assume no obligation to update these forward-looking statements anytime in the future, and the company's actual results may differ materially from anything projected during today's call due to the risks and uncertainties to which the company is subject. These risks and uncertainties are described in our public filings with the Securities and Exchange Commission and at the end of today's press release, which you are encouraged to consult. So on April 2nd of this year, we held a conference call to discuss top-line results from our phase 1 study in Pelizaeus-Merzbacher's disease, or PMD for short, a summary of which had been presented by our PI, Dr. David Rowage, on March 31st, also of this year, at the 2012 European Leukodystrophy Association Families and Scientists meeting. During the conference call, we were asked whether we had any plans to increase the visibility of these very exciting observations into the mass media, and to which we replied that we were working diligently with our collaborators at UCSF to have the study data published in its entirety in a peer-reviewed journal for everybody to see. So today, not only have we successfully done so with the publication of the research article in Science Translational Medicine entitled Neural Stem Cell Engraftment and Myelination in the Human Brain, but we have gone on to take one step further by publishing the preclinical testing that supports the PMD clinical trial rationale and that initiative. And that publication, that article is in the same edition of Science Translational Medicine, which is entitled Human Neural Stem Cells Induce Functional Myelination in Mice with Severe Dysmyelination. So today, I'm delighted to have the lead author, Dr. Nobuko Yoshida, VP of Stem Cell Biology at StemCells, Inc., on today's webcast, as well as Dr. Stephen Bach, our collaborator and senior author of the article. Today, Stephen will lead off and will share some of the very key research findings of the preclinical studies. He will be followed by Dr. Stephen Huhn, who will speak to us about the clinical trial results in PMD. Dr. David Rowage, who is the principal investigator on the PMD clinical trial, cannot join us on this company website, as by policy, in view of the fact that he is a researcher at the Howard Hughes Medical Institute, he is unable to participate in such events. And unfortunately, Dr. Nalan Gupta, who is the co-PI and lead author of the article Neural Stem Cell Engraftment and Myelination in the Human Brain, is in surgery and is unable to join us. So let me start off by just giving you a few introductory background comments regarding Dr. Stephen Bach. Dr. Bach received an MD and a PhD in pharmacology from the University of California at Irvine. He is currently professor of pediatrics and neurology at OHSU. He is the director of the Division of Pediatric Neuroscience at OHSU Dornbacher Children's Hospital and holds the Clyde and Elda Munson Professorship in Pediatric Research. Dr. Bach is the recipient of the Young Investigator Award from the Child Neurology Society, a Booger Award from the American Heart Association, and the Javits Award from NINDS. Dr. Bach's laboratory focuses on the causes of brain injury in newborn infants and is a leading expert in white matter injury and strategies to promote regeneration and repair of brain injury in children with adults. So I would ask Dr. Bach if he would provide us with some additional comments on the preclinical studies to which we referred. On the first slide here, you will see just a brief summary of the two papers that appeared today in Science Translational Medicine. And we will lead off with the preclinical research, and a summary of which is that these human neural stem cells result in new functional myelin. We see oligodendrocyte differentiation in the mice, and we see myelin-enhanced nerve conduction. And we've used MRI to detect human myelination in the animal model. And of course, the relevance there is that we have used MRI to detect myelin in the human patients. So without any further ado, I'd like to hand over to Dr. Stephen Bach.
Thank you, Martin. If we advance to the slide entitled Formation of Myelin Around Nerve Fibers, we'd like to begin there by emphasizing what myelin is. So this is the insulation around nerve fibers, and you can see here where I'm pointing that you can see these areas where the cell body of this myelin-forming cell, the so-called oligodendrocyte, is reaching out and touching multiple nerve fibers and wrapping them with this fatty insulation material called the myelin. And so that's absolutely critical for normal neurological function. And without myelin, patients typically have very devastating consequences, as you'll hear about later in this discussion when we talk about some of the Pelizaeus-Merzbacher patients that were tested in the trial. Now here on the right you can see a human neural stem cell. So this is one of the human CNS stem cells transplanted into one of the myelin deficient mice in the study. And you can see how that cell is reaching out and making contact with these individual nerve fibers and beginning to wrap it. Now, on the next slide, you can appreciate that in the animals that these are myelin deficient mice. It's a special animal model in which these animals develop a neurological condition that looks very much like the patients who were transplanted in the trial. So these are animals who have a very wobbly gait, what we would call ataxia. They go on to develop very severe neurological consequences, including seizures, which become progressively more frequent until it ultimately kills the animal. So these ultimately become very fragile animals that are very difficult to handle. So you can appreciate now on the left that in these animals, the white matter, so that's labeled here, the white matter area with no myelin, is completely devoid of myelin. And there on the right, you can see an example of one of several sites where Dr. Uchida has transplanted the human CNS stem cells into the brain of the animal, and after a number of weeks, these cells have fully populated the white matter, and you can see them extensively in brown, making the new myelin. So if we now move on to the next slide, we've come to one of the more exciting findings in the study, which addressed the question whether neurologically impaired animals would accept the stem cells. Would the cells be able to engraft into the brains of these impaired animals and actually make new myelin? And this was really one of the exciting and central questions of the study, because we had no idea at the outset whether the brain would be normal enough and whether the nerve fibers would still be able to be wrapped by the cells, which themselves are healthy, but of course the brain is not. And here you can see a dramatic example in an animal that was already quite neurologically impaired, where you can see the myelin all in white, which is fully filling in the white matter tracts. And now in this graph, actually on the right, you can see that we compared asymptomatic newborn animals with symptomatic animals who were older, called the juvenile animals. And you can see, looking at the graph, that the amount of myelin that's being formed, which is here on the y-axis, progressively increased over time, from early after the transplant to later times. And importantly, the amount of myelin made in the symptomatic animals was equivalent, no statistically significant differences, from the newborn animals. So this provides a very strong proof of concept that the cells are capable of making myelin in neurologically impaired animals. Here you can see the results from our MRI studies. Now, as I mentioned earlier, these were very fragile animals that do not tolerate handling well. So we had to use a very advanced form of MRI technique using high field imaging, which allowed us to image the brains from the animals after they had died of their fatal seizures. And so this particular result then provides the evidence that these cells, which make the myelin, as you can see here in B, can actually be visualized by the MRI. So what you're looking at in A are regions in green that we predict have no myelin, and regions in red that we predicted would have myelin. And then, after obtaining the MRI scan, we confirmed in B that the areas that were heavily myelinated by the human stem cells were in fact the same areas that we were detecting on the MRI. So this is a very important result because it provides us with the reassurance that what we saw in the clinical trial in terms of the MRI signal, we believe is coming from myelinating cells. Now in the final data slide here, you can see that the new myelin is providing functional improvement to the transmission of nerve impulses along the white matter nerve fibers. And so you can see an example here on the left of how this experiment was done. And two so-called recording and stimulating electrodes are placed into the white matter tract. The tract is stimulated and then recorded from, and we can measure the speed at which nerve impulses move along those nerve fibers. And on the right you can see an example of how much higher the speed and the height of that nerve conduction was in the transplanted animals compared to the controls. So and here you see the newborn animals, and here in the symptomatic animals, an even more impressive result. Finally, here a quick summary then of the myelin deficient mouse model. We've succeeded in demonstrating that these cells preferentially make myelin forming cells, the so-called oligodendrocytes, after they're transplanted in either the pre-symptomatic or the symptomatic neurologically impaired mice, and that these cells not only produce structurally normal myelin, but they also produce functionally normal myelin, which rapidly transmits nerve impulses. And using MRI, we have for the first time demonstrated that we can visualize these cells making new myelin, and importantly, we've been able to do that without modifying these cells at all. So although experimentally it's been labs have needed to modify these cells, the cells transplanted into the patients we now believe can the myelin in those patients can be visualized without any modifications to the cells. So a very exciting result. So I will turn the webinar now over to Martin to continue.
Thank you, Dr. Bach. So before continuing, I'd just like to remind everybody we will open up the webcast for discussion and for questions when we finished with the second part of the webcast, which will be conducted by Dr. Stephen Hune. Dr. Hune is Vice President and Head of the CNS program at StemCells, Inc. He joined the company in January of 2007. Prior to joining StemCells, Inc., he was an Associate Professor of Neurosurgery at Stanford University and Chief of Pediatric Neurosurgery at the Lucile Salter Packard Children's Hospital. Stephen obtained his MD degree from the University of Arizona, did his residency in neurosurgery at the University of Maryland, went on to do his fellowship in neuro-oncology at UCSF Medical Center, and has a fellowship in pediatric neurosurgery at Children's Memorial Hospital at Northwestern University. So, Stephen, over to you.
All right. Thank you, Martin. With that very great introduction regarding the preclinical work that Dr. Uchida have achieved in the collaboration, it's a pleasure now to take you through a summary of the phase 1 trial results in PMD. This is almost entirely based on the paper that came out today, but I would like to go through the more key elements for our discussion this afternoon. The primary goal of the trial, as it was a phase 1 study, was to determine and measure safety and tolerability and feasibility. And in fact, we think we've achieved all that at the 1-year time point after transplantation. But another part of the trial, because we were dosing patients with the disease, was an effort to determine whether we could find any signs of donor-derived myelination by MRI, and whether we could, through neurological and neuropsychological assessments, determine any clinical change that could potentially reflect something different than what we understand to be the natural history of the disease. And in fact, in addition to safety, which I'll discuss coming up, we've been able to show MRI evidence of myelination, as well as some very intriguing gains in motor or cognitive function. And so that's what I'll be discussing in the next few slides. So a little bit of background. Pelizaeus-Merzbacher disease, or PMD for short, is a rare, in fact an ultra-rare, hypomyelinating leukodystrophy. And by hypomyelination, what we obviously mean is these children form very little normal myelin. It occurs as a result of a genetic mutation in the proteolipoprotein, or PLP, which is one of the major proteins that constitutes myelin. And without that protein, because of the genetic defect, these children can't produce normal or produce very little normal myelin. It's X-linked, so it occurs predominantly in males, and there's two basic forms of the disease: a classic form, which has onset later in childhood in a more insidious course, and the conatal form, which it occurs usually very shortly after birth. And these children with the conatal form have the most severe aspect of PMD. They oftentimes present within hours of birth with obvious neurological difficulties, what clinicians see as severe hypotonia or loss of muscle tone. As the children grow, we see that they have a myriad of psychomotor developmental abnormalities. They have deviations in their eye movement, which is referred to as nystagmus. They also have vocal cord paralysis and feeding difficulties. So these are very, very impaired children. If you look at the MRI scan of a normal patient, which you see there on the left, what you can make out is the very dark structure of the white matter on both sides of the brain. It's dark because it's compacted full of myelin. If you look at the PMD MRI on the right, what you see is the areas that appear dark in a normal patient are light or bright on the PMD patient's MRI, and that's because again, the density of the myelin is absent. And again, this is what we see when you look at the autopsy specimens of these patients, is this diffuse, underdeveloped, and abnormal myelin because the myelin is not produced by the patient's oligodendrocytes as a result of the genetic mutation. So although it's a very rare disease, we understand that there's an opportunity to study the disease and perhaps inform us about many other similar diseases or other myelination disorders. We had a long process in which to think about different ways of approaching myelination disorders, and PMD has long been recognized as an appropriate human target in which to establish proof of concept for a donor or cell-derived myelination approach. Because the neuronal pool in these patients, the neurons, if you will, are essentially intact, if you can reconstitute the oligodendrocyte population and achieve myelination of the host axons, then you have the ability to produce a clinical benefit. The other advantage with PMD is that because these children can be readily diagnosed early in infancy, it gives us the opportunity to intervene very early in the disease course, which we think has many biological advantages. And finally, because we're looking at a myelination disorder, we can take advantage of the non-invasive MRI measures that assessed de novo myelination. And that would give us a sense of not only donor cell engraftment, but obviously intended biological activity. So there are many aspects to PMD which are well suited to establishing proof of concept for cell therapy for myelination, which not only would be important for PMD, but potentially other disorders. So this phase 1 trial was conducted at a single site at UCSF. The principal investigator, as Martin mentioned, is David Rowich. He was accompanied by a large and very expert team at UCSF, including Nalan Gupta, the pediatric neurosurgeon, Dr. Stroba, the pediatric neurologist, Dr. Barkovich, and Dr. Roland Henry, both neuroradiology experts. The trial involved dosing 4 patients who were anywhere between the age of 6 months to 5 years of age who had, in fact, the diagnosis of conatal form of PMD. And in general, we felt that these children had to present with symptoms before the age of 6 months. The dose that was administered was 300 million cells, or 75 million cells in each of one of 4 sites. They were treated with immunosuppression for 9 months post-transplant, and then they were subjected to the routine safety and other examinations, which included neurological assessments, neuropsychological evaluations, as well as MRI and the different MRI techniques to assess for myelin, which are referred to as diffusion tensor imaging. After the phase 1 study, all the children are going to be followed in a separate 4-year long-term observational study. So this just shows the transplant paradigm for where we introduce the cells into the brain. Given that this is a first-in-human study, if you will, for cell-based myelination, we had to pick the safest and largest white matter structure in the brain, which is sometimes referred to as the centrum semiovale, or essentially the white matter of the frontal lobes bilaterally. There were 4 sites picked within each brain, 2 sites in each frontal lobe. So this shows the demographics of the subject at their baseline. They ranged in age from 16 months in the first patient to 66 months of age in the 4th patient. The youngest patient was 14 months of age, and that was subject 3. As you can tell from the table, 2 of the patients had tracheostomies and gastrostomies almost from the time of birth, which reflects, again, the severity of the disease. None of the patients had the ability to self-feed, and none of the patients had the ability for independent ambulation, although the 4th patient had some degree of ambulation, but with significant support. All the patients were alert because their neuronal pool is largely active, and all of them had significant language deficits, both with expressive and receptive, although patient 2 and patient 4, being slightly older, had some primitive language function. In terms of safety and tolerability, we recorded 54 adverse events for the trial with the usual suspects of fever, rhinorrhea, rash, and minor GI disturbances, all of which were related to the underlying disorder. There were 2 serious adverse events, 1 tracheitis and 1 fever, again, None of these events were attributed to the cells, and none of the patients developed any seizure activity during the course of the year following transplantation. And more specifically, from a safety perspective, we could detect no focal or global loss of neurological function, either immediately following the surgery or thereafter. So this is a summary of the neurological examinations for the patients. And I've taken them, basically, all four are described in the paper. But to review, subject 1, who was 16 months at the age of transplant, basically remained neurologically stable. We detected no particular gains in that subject, although the clinicians recognized that his nightly continuous positive airway pressure had decreased. Subject 2 had improved truncal support, and he actually had the ability to take steps with assistance at the end of 12 months, and he began speaking audible single words with the ability to follow two-step commands. Subject 3 developed upper extremity anti-gravity strength and started taking solid foods orally, and he also had reduced continuous positive airway pressure at night, which is a sign of some respiratory integrity. And subject 4 had improved truncal support and progressed from use of the walker at baseline that required significant support to walking with minimal assistance at the end of 12 months. That subject also developed the ability for self-feeding and to follow two-step commands. And I think it's important to acknowledge that this is an open-label study, and any results, any clinical outcomes should be interpreted in that light. And we also recognize that further clinical investigation with proper control subject is required to confirm the clinical benefit from any intervention. But I think it's fair to say that measuring gains in neurological function in the context of what is a severe and progressive neurodegenerative disorder has been a particularly intriguing outcome of our study, and we think it suggests a deflection from the natural history of conatal PMD. This slide shows the neuropsychological outcome, which is the Bayley scales of infant and toddler development. This is all measured in raw scores, and as you can tell, subject 1 in particular had very, very low raw scores, in part because of his young age, as did subject 3. In subject 1, basically, you can tell from cognitive, language, and motor skills that overall no significant real trends there. But as you look at subject 2, 3, and 4, you can see that in some cases, and select subsets of the Bayley, that there's been some measurable improvement in those areas, particularly in subject 3 is noted there. So in addition to their neurological assessments and their neuropsychological assessments, which I've just gone through, the patients also underwent routine MRI examination. And the routine MRI examination involved not only the morphological or conventional MRI images, which you see here, but as well the diffusion tensor imaging, which is a more sensitive way of looking for any signs of myelination. What's shown here is in patient 2, pre-transplant and post-transplant images. They're referred to as T1 and T2 images, which depict small areas of white matter changes near and around the area of transplantation. And in this patient, you see that by the red arrowheads. These changes were seen in 2 of the other subjects as well, and they did not correspond to any artifact that we saw in the exams, and they developed and progressed with each examination. The other aspect of the study involved the diffusion tensor imaging, which is a technique that measures the magnitude and direction of water molecule movement that's bound to myelin, which you can see there in that schematic. The MRI has the ability to measure the movement of these water molecules with magnetization, and there's a very specific pattern that's reflective of myelin and water movement, and that can be then illustrated and demonstrated either qualitatively, as you see there by the mapping image, and quantitative, which are the images that I'll go to toward the end. So this is a very sensitive method, a non-invasive method, if you will, of examining whether there's been any de novo myelination changes. It's used in conventional imaging for patients who have MS and other demyelinated disorders. So it lended very well to our study to determine whether we could, again, through very sensitive techniques, measure changes in myelination in the transplanted regions. This just shows how the DTI analysis was conducted. And we had the control regions, which were areas of white matter which we considered remote to where the cells were transplanted in the frontal lobe, as well as the transplanted regions of interest in the exact frontal lobe white matter that was transplanted. So the DTI was conducted in all of those areas and then analyzed. What you see here is the qualitative DTI mapping in one of the patients through actually 18 months of data. And this is an axial image of the brain on the left, and you see the orange and yellow, which signifies some primitive degree of myelination in the white matter in this patient, but a normal subject would have far greater color and signal. You can see there on the 6-month that there's not been dramatic changes between the pre-transplant and 6-month color mapping by DTI. And again, this is a qualitative measure. But as I scroll through the 18 months, you will notice in the area where the cells are transplanted, again, the right frontal lobe matter, that this signal will change and evolve. And I'll click through these now. So we see an increase qualitatively by DTI in the area where the cells were transplanted that was not observed in the non-transplanted areas. From a quantitative standpoint, what you're looking at now are the percent changes, both absolute and relative, for all 4 patients across all 12 months of the study. So the images that I just showed you were qualitative. This is the quantitative data of each of those patients. The top 4 panels shows in red the transplanted percent changes, or the transplanted regions of interest, versus the control regions, which is in blue. And in each case, you can see in red, the transplanted regions has an increase in absolute change, one of the parameters of the DTI over time. It's important that we see that this change is progressive, it's slow, but it evolves with each subsequent MRI. And more importantly, it persisted after we stopped the immunosuppression at 9 months. If you look at the lower 4 images, what you see is the relative change in the transplanted regions compared to the control regions. And again, in 3 of the patients, you see an increase in relative signal change by DTI in the transplanted versus the control. In patient 3, which is the 3rd one from the left, you see that there's been some variability there. And we think that the young age of this patient probably reflects a degree of maturational changes that may be going on in the background and that would contribute to that variability. So in summary, we have conducted this phase 1, and we're very pleased with the favorable safety profile that this intervention has resulted in a very feasible and well-tolerated treatment. We see signs of clinical changes that we believe suggest a departure from what we understand to be the natural history of the disease, and that the MRI findings are consistent with de novo myelination in the regions that we transplanted in all 4 subjects. The phase 1 data strongly supports the implementation of a controlled phase 2 trial in PMD, and the MRI data also is very suggestive and supportive of beginning to explore other myelination disorders, such as certain forms of cerebral palsy, spinal cord injury, even transverse myelitis, and multiple sclerosis. In closing, I think as a clinician, we are, and part of the team, we're very pleased with the outcome of the study. And we're very excited about taking the next steps in PMD, as well as I mentioned, exploring the other potential for this in other myelination disorders. We very much feel like we're on our way to exploring the regenerative potential of neural stem cells and possibly helping patients who suffer from these very devastating neurological conditions. And we think the data from the preclinical studies that Dr. Bapp went through and the results that we've now seen in the human trial have really indicated we've accomplished this first step.
Thank you very much, Stephen. We really enjoyed listening to very exciting results of the study, and it's great that we're now finally able to share data and put all of this data and this information out into the public world for comment and for scrutiny. So if there's anybody who has joined the webcast, and if you would like a question, if you would like to pose a question to Dr. Hune or to Dr. Bach or to the company, would you please let us know through the chat room, the chat box, and we can enable that question.
How we're going to define core, but like a core set of
So, again, this is Martin.
But the reason I'm bringing it up is to say, because I always feel like, and it's felt like also, that we're always trying to catch up on something, either having to deal with different things.
Hey, Joe? Joe? Joe, could you come back online, please? Because we didn't get your question. Could you come back online and repeat it, please? Hello, Stephen.
I'm here, Martin.
Yeah, Stephen, do you have a question? Stephen Dunn.
Yes, I do, actually. First of all, congratulations to the entire team and for both research teams. I guess it's the first time I've ever been on a call where we discussed the preclinical results and the clinical results all at the same time. So what I'd like to put out to both Stephen Bach and Stephen Yoon, when we look at the results from the murine model and what we saw, do you guys believe that the results that we saw in the PMD in the children trial were consistent with what we expected to see based on the preclinical model? In other words, were there any surprises moving from the preclinical model to the actual human results?
So I think it's a good question, and we're essentially what we saw in the preclinical work with Dr. Bach and Dr. Uchida was evidence that the cells differentiate into oligodendrocytes and then myelinate the host axons, and they can detect that by MRI. If you think about it, we've essentially see replication of that result in the human study. They see it in pretty much every animal that they've dosed, and we see it in all 4 patients in our study. So in many, many ways, there is a straight parallel between the data that we've seen in the preclinical study and what we're now starting to detect in this phase 1 study. So it's a natural evolution of the data, and for me suggests that maybe preclinical data with stem cells, with neurostem cells, might be more translatable into humans.
Okay. On the PMD trial, it looks like we didn't really start to see any myelination until 6 months or maybe even after. Do you think the impact of immunosuppression kind of suppressed the myelin development until after that immunosuppression was stopped? Because again, in the mouse model, we didn't have any immunosuppression. So do you think that might be why we didn't start to see anything until after 6 months?
Well, the mouse model doesn't require immunosuppression because it's an immunodeficient mouse.
Right.
And as best we understand the interaction with immunosuppression with our cells in preclinical studies, we don't see that it inhibits the development of oligodendrocytes. I think what's more important about the DTI data, the MRI data, is that it's a progressive and slow evolution. And if we were seeing things within a month or two of transplantation by DTI, that would be okay. But biologically, we think it takes some time for the cells to engraft, to potentially migrate, and then to differentiate into oligodendrocytes and then myelinate. So that all doesn't happen probably within weeks. That's probably months. So the radiology physicist, Dr. Henry, is very pleased that he sees a smooth signal building in each of the patients, and he worries about signals that are very noisy and that perhaps come on too early. So I think it's the smoothness and the progressiveness of the signal, and the fact that, again, that we see it persist and continue to evolve after we've stopped immunosuppression. So all of that is very supportive that the cells are, we think, myelinating host axons, and that's why we use the word progressive and durable, because it builds over time and it persists after the immunosuppression has been stopped.
Okay, yeah. I guess I just want to put a finer point is I've been active immunosuppression between the two trials. So that said, I don't know if, do you feel we're starting to see results that are based on the age of the patient? In other words, do you feel that let's say the older the patient, within reason, we're probably seeing a better result, or if they're, let's say, 14 months or younger, that it might not take as well, or do you think that's just an extrapolation that is undeserved?
Yeah, I think we have to be very careful to draw correlations with only 4 subjects. But if you look at the youngest subject in the trial, subject 3, and again, it's in number 1, that he actually had the most highest absolute DTI change in the region of transplantation. And we're still trying to understand these diseases biologically, but it's possible that the developing brain, the 14-month-old brain, is more favorable. But then if you look at the results that we see now in the patients who were between 3 and 5 years of age, so these are children that have had PMD for several years, we still see evidence of a DTI change in that more mature brain. So I think, Stephen, we're going to learn a lot more about this as we enroll more patients and look at different age ranges and as they enter the trial. It's difficult to draw correlations with only 4 subjects.
Okay. Final question. I'll jump back in the queue, but it's multi-part. Dr. Back, do you feel there's more to be done in mouse models, or do you have plans to do more work, and what are you looking for if you are? And I guess for Martin and Stephen Yoon, what's next? Do you feel FDA, a PMD phase 2, do you feel FDA is feeling much more comfortable with your human neuronal stem cells? Because we have not seen any safety issues in any of your human trials. So for Dr. Back, what's next, if anything, in animal models? And for Martin and Stephen Yoon, what's next? And do you feel FDA is feeling much more comfortable? Safety.
So, Dr. Back, would you like to answer the first part?
Certainly. So, I think that the road forward looks fairly clear for leukodystrophies like PMD. And I think these studies provide proof of concept that a clinical trial would be indicated. And so, but I think what is exciting about these results is the potential to extend these, as we discussed earlier in the webinar, extend the application of these cells to other human disorders, there's a wide variety of disorders of human myelin disorders that could be studied at the preclinical level. My lab, for example, is particularly interested in white matter disorders in children and neonates, and I think we're particularly keen on knowing what are the particular challenges for getting cells to engraft in injured brains, as you see, for example, in cerebral palsy or multiple sclerosis or even the aging brain. And so there's certainly plenty of work to be done preclinically to expand the scope of applications for these cells.
Okay, great, thanks.
Mm-hmm.
So Stephen Dunn, that is, we have a lot of Stephens on the call today. So Stephen Dunn, in answer to your question about what's next in terms of the PMD program, well, yes, you're correct. Your observation is correct that to date there have been no issues, no safety issues have arisen with regards to the cells or the immunosuppression or the procedure that we use to deposit the cells, whether it's into the brain or the spinal cord, and recently into the eye. So obviously, as the safety profile is built out and folks at the FDA and clinicians and patients are gain a greater degree of confidence with regards to the safety components, then the prospects are good for us in terms of advancing with our clinical development programs. With regards to PMD, we certainly, with this data, are very intrigued with the results, and we're enthusiastic and we're encouraged. And we will be engaging with the FDA to talk about a study that would be of a control design nature, a phase 2 study in PMD patients. With regards to other leukodystrophies, of course, we're going to take that under advisement, and we will be speaking to expert scientists and clinicians as to other possible targets that are leukodystrophy in nature.
Okay, great. Guys, great background call on both papers. Congratulations to both teams. Congratulations to StemCells, Inc., and looking forward to an exciting 2013.
Thank you very much, Stephen. Are there any further calls? Any other questions? Okay, looks like we're good to go. Thank you all again for joining us today. We really are very excited about these papers, and as Stephen Dunn said, it's his first time that he has actually had the opportunity of looking at all of the preclinical data and the clinical data in a back-to-back peer-reviewed journal. It doesn't happen all that often, and we're very proud of that. And as you said, we have great scientists and great clinicians working on these programs in harmony and in collaboration, and we thank them all very much. Thank you all.